You're listening to Treating Blood Cancers, the LLS podcast series for professionals. We will be joined by cancer experts to discuss blood cancer diagnosis, treatment, side effects management, and the importance of clinical trials. They will share their experience in treating patients and discuss strategies for optimal patient care. Let's get the conversation started.
Welcome to Treating Blood Cancers, the LLS podcast series for professionals. I'm Dr. Ken Miller. I'm a medical oncologist and an LLS volunteer. And I want to thank you so much for joining us on this episode to discuss highlights from the 2024 ASH, American Society of Hematology annual meeting. Today, we're joined by Dr. Lee Greenberger, who's the Chief Scientific Officer and Senior Vice President at LLS. Lee, thanks for joining us.
My pleasure.
So I want to start out with a question. I think we've done this as a series for several years. So I'd love to get your view from just the 60,000 foot level. How was this meeting in terms of level of excitement, level of advance, compared to the many meetings you've been to?
Yeah. So I would say that this meeting is on the same trajectory as in the past few meetings. We're pushing the progression-free survival intervals, longer periods for many different diseases, including myeloma, particularly lymphoma. And we also have a couple of new drugs for leukemia. So I don't think we saw anything like knock it out of the park, like, wow, CAR T, what is this? BTK, what is this? Almost a decade ago. But we're seeing now all these agents used in combinations and second, third, even fourth generation drugs pushing the progression-free survival and hopefully overall survival out further.
So, which is, and by the way, exciting to hear. I'm a community practitioner, and the sense of all these things happening in multiple diseases is really exciting. Let's start out with, because you mentioned myeloma, there was an abstract. This was Hansen about CAR T and myeloma. Tell us about that.
Right. So, first of all, we're seeing definitely an increase in progression-free survival in myeloma patients with multiple therapies. We're also seeing actually an increase in patients who are reporting with myeloma. There's 35,000 new cases of myeloma, and that is on the rise, and it's significant. Okay, so now the question is, what are we going to do about it? How are we going to treat myeloma? And one of the ways, which was really started with their first approval of CAR T, this is an immunotherapy where you basically take the T cells, re-engineer them, put them back into the patient. They attack the tumor cells. That was first developed for lymphoma. About 3 or 4 years ago, the first CAR Ts were approved for myeloma. That is, in particular, cilta-cel as well as ide-cel, two approved products, BCMA, which is on the surface of myeloma cells, BCMA CAR T cells. We have multiple years of experience with these compounds, with these living drugs, if you will. And so cilta-cel is compared with ide-cel in this abstract by Hansen. And basically the punchline is cilta-cel performs better in terms of efficacy versus ide-cel. There is some more toxicities with cilta-cel, but from an efficacious point of view, it looks more efficacious than ide-cel. So it might be the CAR T of choice, albeit there's some toxicities, which we know about, the neurotoxicity, the immune activation, the so-called CRS phenomena, which can be, for the most part, managed.
I wanted to ask you about that because we have a lot of abstracts on CAR T. Again, from a 60,000 foot point of view, what was your view at this meeting in terms of managing toxicities and administering CAR T as opposed to what it was several years ago?
So I still think that we're definitely getting a certain rate of grade 3 events that are serious, basically telling us this therapy is not ready for communities, that's for sure.
Yeah.
And we have to still be careful, even on an outpatient basis, where this patient may get the cells and say, okay, go back into your neighborhood or go to the hotel, waiting side by side. But you need to be on standby because these events could happen. They typically happen shortly after the CAR T administration, but not always. In terms of controlling them, I think there are more CAR T centers and people are using agents to control, for example, CRS, even by giving tocilizumab prophylactically, even before to actually even cut down the CRS. So that's some of it. The neurotoxicity still remains difficult to control. And another side effect called HLH, which is a more dramatic side effect, immune activation, that's still a problem. We really don't know how to control that. Doesn't happen too often, but it can. Bottom line is, CAR T therapy, at least a BCMA CAR T for myeloma, works well. I'm going to qualify that in a moment. And it's an option of choice after first line failures, or actually initially approved after multiple failures. For the multiple failure patient, let's say pentafailure, the survival curve is just going to continue to go down. We don't get cures for those patients. For the after the first line failure, we don't know yet, but it's looking those patients are much better controlling the disease. The progression-free survival rate is higher. We'll see whether we get some cures in those patients. But all together, even with the toxicities, we're sort of marching towards longer progression-free control and maybe some cures in myeloma. We're certainly getting cures in lymphomas and leukemias. Myeloma has been more difficult.
Got it. So I have to say there was a very interesting abstract that I'm eager to hear about. This was number 733, Demopoulos, treatment of pre-myeloma with drug therapy or diet. So tell us about that.
So actually now there are sort of three important advances that are emerging. So smoldering myeloma can convert to myeloma. There are high-risk patients. I think the 50% conversion rate for those patients is about 1 to 2 years for high-risk smoldering myeloma patients. Those patients typically are sort of watch and wait. But now with the advent of daratumumab, CAR T, bispecifics, and now even diet. The concept is, can you control these diseases and block them, intercept them from converting over to myeloma, thinking that this is the lowest grade disease. If you block conversion, maybe you'll even block it permanently. You'll never get myeloma. That's sort of the concept. So what Demopoulos and colleagues did was to use daratumumab, which is of course approved for myeloma itself, and compare that with a watch and wait phase 3 comparator trial and ask, does daratumumab reduce the incident of conversion to myeloma? And the answer was it does.
Mm-hmm.
Now, the question is, with that therapy, it does have some side effects. It's an expensive treatment. And so for a smoldering myeloma patient, the question is, do you go ahead and treat with daratumumab, or maybe these smoldering myeloma patients, even of high risk, will never convert to myeloma? How do you know? So that's sort of the dilemma with treating a watch and wait patient or a patient that doesn't have full-blown disease. Switching over, another option. Here's the more aggressive option. We talked about CAR T. Irene Gobriel at Dana-Farber is actually using CAR T therapy in smoldering myeloma patients to see if she can ultimately block conversion, but
Yeah.
just as an immediate endpoint saying, can you reduce the detection of the disease by minimal residual disease, MRD? Are the patients becoming MRD negative? They just got 6 patients. They all become MRD negative
Wow.
in smoldering myeloma,
Mm-hmm.
suggesting that we're going to block that conversion.
Yeah.
And then the final one was to use a diet, a high-fat diet in smoldering myeloma patients. Was done in 20 patients, was also done in mice, actually, as sort of a preclinical proof of concept. You could block it, you can block conversion in mice or reduce the impact of myeloma. For the smoldering myeloma patients, these 20 patients saw an increase in time to progression, to conversion of myeloma. So diet is also possibly another way to control disease. This isn't, by the way, the first example of diet to control a blood cancer. This is so-called, I think it's neutral intervention for smoldering. It's now going to be attempted to be done in myeloma
Yeah.
to reduce the progression of myeloma. That's number one. And number two is Marco Ruella at UPenn has treated patients with, or has looked back and say, what happens if you switch the diet on patients that are getting CAR T? does it have a better outcome in terms of treating lymphoma? And the answer looks to be yes. This was actually done looking at diet in mice and then looking at the active ingredients in mice and then going into patients and saying, is that active ingredient more present in patients that responded well to CAR T therapy? And the answer is yes. So the
Wow.
suggestion is diet may actually possibly heighten the immune system, but the end result is better control of disease, whether it be disease before you actually get full blown disease or even advanced disease.
So, by the way, I have to say, patients ask regularly, what can I do? What should I eat? And typically the answer has been just a regular diet, a healthy diet, etc. This is fascinating, the idea that perhaps diet can modify response. So we'll wait to hear more.
This is all sort of also interwound in the concept of controlling the bacterial content in the GI tract can also influence the immune system. And the big picture is, I mean, the big statement in the last 10 years is the immune system plays a major role in terms of either allowing tumors to progress or hyperactivating them to control the tumor.
Let's digress for a minute because I think this is fascinating. And even for me, the concept that people sometimes can live with minimal residual disease and not progress, which is probably just echoes why do some people develop certain malignancies and others don't. Your read on the literature, is this humoral immunity? Is this cellular immunity? Is it all of that?
Yeah, I don't think people really know for sure. I
Yeah.
think it could be both. It still really it requires a lot more exploration. And even the high fat diet that's been used to block smoldering myeloma conversion to myeloma phenomenon is there. What the ingredients are and whether that's, which components of the immune system are really being activated and what's really controlling the tumor, not really known.
Yeah, yeah, interesting. All right, more to follow in years ahead. Let's switch to leukemia, acute leukemia. Last year, we talked some about menin inhibitors. So, what was new this
Right.
year?
Right. So certainly the first menin inhibitor, revumenib, got approved by the FDA about a month ago. And so what we've gone from, and that's number one, got a menin inhibitor on the market to control relapse refractory AML for patients that have a certain mutation. It's called K2M2A. That's what the approval is for. Okay, so that's first piece. There are now multiple menin inhibitors, about 5 or 7 companies working on menin inhibitors. So we're going to see a flurry of these menin inhibitors that are likely to get approved. Okay, that's the second point. How well do they work? Well, they work about as well at this point as many of the monotherapy agents that have been approved, the FLT3 inhibitors, the IDH inhibitors. The complete response rate so far is kind of like in the 20-30% range, complete response rate. It's not knock it out of the park. And in some cases, the overall survival is increased. Granted, in a relapsed refractory patient, it's a small amount, 6 months. That's where we are with AML. We got a lot of work to do in AML, and that's why LLS is heavily invested in AML.
Yes.
I'll digress in a moment about the story between menin and LS in a moment, because that's kind of fascinating. Okay, so that's number 2. Number 3, menin inhibitors for NPM1 mutations and menin inhibitors in combination, point 3A and 3B. The NPM1 is mutated in about 30% of AML patients, whereas K2M2A is a rare mutation under 10%. It's highly prevalent in infants, and by the way, the drug's not approved for infants yet. But the point is, if the drug can be used in NPM1 mutants, that'll expand the utility of menin inhibitors. It's likely still not going to be knock it out of the park, and the drugs are going to have to be learned in the clinic to use them in combination. Those studies are ongoing. There are 3 companies now already that have the menin inhibitors of the ones that they own in combination with 7+3, venetoclax, azacitidine, so the latest and greatest of how we treat AML. So that's all sort of coming. A little bit about the history of menin inhibitors, which I must digress because
Please do.
LLS invested in menin inhibitors goes back to about 2005.
Mhm.
We're now, think about this,
20 years.
20 years from the concept of, hey, menin interacts with MLL and that can cause leukemia in animal models, all the way to Yolanda Grambecka and her husband Thomas Cherpecki saying, hey, there's a small molecule inhibitor that can inhibit that interaction. Then got picked up by biotechnology companies, clinical trials done. We're now 20 years downstream. LLS is invested all the way along through Yolanda Grambecka, through Cleary at Stanford to make the initial observations. So we follow this thing all the way through, including our venture philanthropy program to assist these molecules through. So for LLS, I mean, like, it's a knock it out of the park. It's like, wow, we stuck with it for 20 years.
Yeah, yeah, excellent. Wow. So I'll tell you, I hope there'll be more of those. I'd love to, 20 years from now, I have a feeling you and I will be quite retired. But
It's true. And, I think this just shows, even with CAR T, when Carl Jr. was learning how to grow T cells in culture back in the 90s, which we funded, those early investments need to be done. We need to understand how these diseases work. Then we need to make the small molecules or the immunotherapy. It takes decades.
it does. There was another abstract that I found interesting for patients that are perhaps less fit or less able to go through induction. There was a paper by Goldfinger on venetoclax and decitabine. What can you tell us about that?
Yep. So I know they work well because actually we gave some additional funding to Montefiore Hospital to run those studies. So here's the concept. The so-called VIALE-A study, which is azacitidine venetoclax in combination for the treatment of AML, is basically at this point standard of care, particularly in patients that are not capable of getting the intense chemotherapy of 7+3 and also ineligible for transplant. And the combination works fairly well. Right now, the label for venetoclax requires that the drug be given for, I think it's 28 days or a 28-day cycle. It's given intensively and we're on multiple cycles. The problem is many of those patients can't even take the venetoclax that long. The side effects are too dramatic and they don't even get the 28-day dosing. Nevertheless, that's what the label says, and physicians are prescribing by the label. Some of the tertiary centers have now backed off on venetoclax. They'll give 2 weeks of venetoclax. And so the question is, what is the optimal dose of venetoclax? So what Goldfinger did was because it was COVID and they didn't want patients coming in to get their treatment, they said, you know what? Take venetoclax once a week with decitabine once a week, and we'll manage it that way. And it turns out to be a pretty effective way
Yeah,
of
yeah.
managing AML. And so the question is, can we reduce in a large study show that you don't need venetoclax 28 days, you can do 2 weeks or perhaps even once a week and avoid a lot of the toxicities and still retain the good efficacy?
I'm sort of shaking my head here because who would have imagined, years ago, patients taking pills and doing it at home? It's just incredible for that disease. The CAR T has moved into the AML space as well. What was discussed at the meeting?
Yeah, so there's multiple shots on goal for CAR T for AML. We know so CD33, which is the target of gemtuzumab ozogamicin, that's Mylotarg, one target. CD123, another target. And then I could rattle a whole bunch. Those are the ones that have been really the most intensively studied. Here's the problem with CAR T for AML. You can not only kill the AML cells, but you kill the normal stem cells. So there's a very small therapeutic window. What happens is you might get some disease control, but you also get tremendous hematosuppression,
Yeah.
prolonged hematosuppression. In fact, some of these patients need to go to a transplant because the hematosuppression is too dramatic. So CD33 and CD123 CAR T for AML is still really problematic. Are there going to be ways around this? Are there other CAR Ts to different targets that may have a wider window? Coming. There's some companies that are pursuing other targets for CAR T. There's one for CLL1 that the folks at Sloan Kettering have done. And so actually, what this is designed is, first of all, do allogeneic, take the T cells from a donor. In other words, so, okay, the T cells are not as exhausted and beat up with all these chemotherapies in the patients. Take that. In the case of CLL1, use an IL-18 booster, which actually Rainier Brenchen did that in mice about 10 years ago, and somebody finally picked up and brought it into the clinic. So IL-18 to boost up the activity of these T cells, CLL-1, put it into patients. Sloan has put it into 5 patients. 3 patients have a leukemia-free state, still have to go on to transplant for some of those patients. So new targets coming, CAR T for AML still a challenge.
Yeah. To be honest, sounds complicated, but hopefully worked out over time. What is CLL1, Lee?
CLL1 is simply just a protein that is expressed on the surface of AML cells, preferentially expressed in AML cells. It's
Got
not
Okay,
CLL, by
good.
the way. I'm talking about
Y-
CLL1, distinct entity, protein on the surface of AML cells. Yeah.
Got it. I wanted to ask you a disease that I've seen very little of, but is CMML. Tell us about anti-GM-CSF. What is that, and how does that fit into therapy for this rare disease?
So CMML is an area that LLS has been intensively interested in. We have a big program in it. Just to kind of give the audience of what is CMML, it's a leukemia. It's a rare one, about 1,000 new patients a year. Disease is treated principally with HMAs, azacitidine or decitabine. Unfortunately, it's been studied already. Decitabine compared to hydroxyurea, no difference in terms of outcome. Overall survival about 30 months, event-free survival about 18 months. These patients don't do well. 30% of these patients convert over to AML. The outcomes are really poor, high unmet medical need. Okay, so maybe about 5, 7 years ago, Eric Padron and Ronald Padnaik used anti-GM CSF antibody called linzoluzumab to show that it might have some activity in CMML. There is now a study ongoing in Australia using lenzilumab, anti-GM-CSF antibody, in combination with azacitidine, and getting much higher response rates on the order of about 50% complete response rates, which is superior to historical rates of about maybe 10 to 15% with azacitidine alone. So it looks like among the most promising therapies for the treatment of CMML. By the way, these patients that got treated had to have a RAS mutation. RAS pathway mutations are fairly common in CMML patients. So where do we stand? Response rates look good. We'll find out more about duration. Probably going to require a much larger study, maybe comparing it with azacitidine head to head to show that anti-GM-CSF in combination with azacitidine is better than azacitidine alone. And if that works out, we'll have one of the first new therapies for CMML in decades.
Yeah,
That all
terrific.
said, there's many different agents that are being pursued for CMML, some which have failed, some that LLS is involved with, about 6, 7 new therapies on the way in development for CMML, understudied area, but which looks promising with multiple therapeutics and really simply just understanding the mechanism of the disease. We need to do that.
There was an abstract by Kim, number 343, about splice proteins. Yeah, tell us
Yeah.
about that.
Yeah. So it turns out that when proteins are made, RNA encodes the proteins, and pieces of RNA have to come together. The so called exons come together, and that makes the protein. This is all controlled by you remove the so called introns between the exons, and they get spliced out to make the protein. Well, there are multiple genes that control splicing. There are splicing mutations that are very common in CMML. About 50% of the patients have splicing mutations. They also occur in MDS as well, and they're also observed in something called clonal hematopoiesis. It's sort of a pre-leukemia state. Okay, so now what Omar Abdel Wahab did at Sloan saying, well, if the splicing mutations are impaired, I should be getting aberrant proteins or pieces of introns or aberrant splicing that will create abnormal proteins. In other words, a protein that we've never seen before, so-called neoantigen. So if those novel proteins are expressed on the surface, I may be able to make an antibody to those and to recognize the so-called neoantigen and kill the tumor cell. That's the concept. And so that's what they're doing.
So, any idea about efficacy there? Are those in clinical trials?
Still too early, still coming. But the concept of there's a novel protein that's abnormal that is going to be unique to the tumor cells that you could use for targeting for CAR T therapy or bispecific therapy. That's kind of the gist of it.
Terrific. We have a lot to talk about in non Hodgkin's lymphoma. So let's go right to it. Let's talk because there, unfortunately, in the community, there's a lot of diffuse large B cell lymphoma. So, what was new and exciting there?
So, first of all, big picture, CAR T is a treatment for relapsed refractory DLBCL showing nice efficacy. For Rituxan based therapies, frontline, about 50%, maybe 60% of those patients are going to get long term disease control. It's those rest of those patients, the 40% over, that are going to need some additional therapy. And so immunotherapies, whether it be CAR T or bispecifics, have really basically been integrated now into the treatment of DLBCL as well as a whole bunch of other lymphomas, to the point now where we can even say, gee, instead of using so-called R-CHOP or R-EPOC, what if we just use a bispecific antibody and use it right up front and compare that, for example, ultimately with R-CHOP? Now, the comparisons haven't been done yet for DLBCL, but there's an abstract by Morshauser, which is used epcoritamab, which is a CD20 by CD3 bispecific. Basically, CD3 attaches to the T cell, CD20 attaches to the tumor cell, bring the T cell to the tumor cell, kill the tumor cell. Been approved for relapse refractory. Let's now use it frontline, right? And in that study, in anthracycline ineligible patients, that's what's been done. 44 patients have been treated, complete response rate 64%, 6 month progression free survival 84%, 14 out of 14 complete response MRD negative. Looks pretty promising.
Sure does.
So the concept is, gee, maybe we can skip the chemoimmunotherapy and go right to a bispecific, or perhaps, go right to a CAR T therapy. CAR T, little more complicated, maybe higher rate of CRS or ICANS you got to worry about. Bispecifics are off the shelf. You don't have to manufacture the cells, go right from the vendor right into the patient. Don't have to do any sort of pre-treatment of the patient with chemotherapy for CAR T, go right into the bispecific. So pretty attractive alternative coming. I guarantee that's what we're going to see
Wow.
in the next few ASHes as to how well these immunotherapy works right up front.
Yeah, I mean, I have to say that the listeners can't see it, but I've got a big smile on my face because, again, picturing, we've used R-CHOP for years. We haven't really been able to move the needle that far with it in any way, but that would be terrific. Wow. Okay, something to look forward to.
I should qualify that. What's the long-term consequences? What's the long-term outcome with, let's say, bispecific antibodies? And what happens if you fail the bispecific antibody? What do you do after that? This concept of what is the sequence of treating patients
Yes. Yeah.
with all these different therapies needs to be defined. And that's really going to be done by brute force clinical trials to understand that.
Yeah, yeah, thank you. We'll go to the other side of the spectrum, or at least a less aggressive disease, follicular lymphoma.
Right. So follicular lymphoma, many alternatives. Of course, for the relapsed refractory follicular lymphoma patients, the so-called ZUMA-5 study, CAR T therapy, CD19, 5 years out now, median PFS 57 months, and the survival curve is flattening out, right? Suggesting that we're going to get, for follicular, long-term disease control, maybe a cure, right? And of
Mhm.
course, with follicular, we know that follicular patients can have relapses. The relapse can occur 5 or 10 years later. And so, how do you know when you actually have a cure? What's the definition of a cure? I got the therapy, I'm no longer on any therapy, and the disease is not detectable. Do you wait 5 years, 10 years? When do you declare victory? But the Zuma 5 study suggests we're sort of on our way. There are additional therapies, alternatives. There was a paper presented by Sen. This is a late breaking abstract presented at the end of the meeting, if you wanted to stay till Tuesday. This was tafasitamab, that's a CD19 antibody combined with rituximab lenalidomide. So Rituxan is anti-CD20, tafasitamab is CD19. So use tafasitamab in combination with so-called R squared, rituximab plus lenalidomide. What's the outcome? In those patients, phase 3 patients, 548 patients, big trial, progression-free survival rate, 22 months versus about 14 months in favor of tafasitamab, rituxan, R squared combination.
Right, right.
Let's go back. Now, that's relapse refractory. Now, let's go to first line and let's go to the watch and wait patient. Let's go try pushing these therapies back, right?
Yes, thank you.
Yep.
Important topic.
So, mosunetuzumab combined with polatuzumab, that's an antibody drug conjugate in patients with high tumor burden. 83% complete response rates in those patients. So CD20, mosunetuzumab, CD20 by CD3 bispecific looks promising in those patients. And let's go even further back. Now, watch and wait patients, right? Same concept. Can you block watch and wait patients from needing full blown therapy? How are you going to do that?
Yeah,
And so, one way that's being explored is using a peptide mix. It was developed by a company, Enterome. Actually, LLS has supported this company through our venture philanthropy arm. A peptide mix given to patients who are watch and wait to see if you can reduce disease in those patients. And so far, only 13 patients have been treated, but 6 patients have either had a partial response, one patient's actually had a complete response, suggesting, again, same type of thing peptide mix, immune activation, control early stage disease. Sound familiar, right? From
it does. Sounds
myeloma.
like myeloma.
Yeah, right.
Yeah,
Yeah.
yeah,
So that's coming. And so we'll see how that works out. There's going to be a bigger study that's going to be done with that. But can you imagine that instead of treating with the intensive chemoimmunotherapy, we should be able to control long term watch and wait patients? They'll never get follicular lymphoma.
yeah. So the idea of a patient, in a sense, outliving their disease is terrific. And actually, in a sense, outliving our retirement as clinicians.
Right, right, exactly. And so it's interesting. LLS has done a look back over the 75 years, how much we have increased life in those patients. And the answer is about 21 million life years saved over the last 75 years. What does that mean? That means that basically, as a composite, all patients were gaining millions of life years. And Rituxan, by the way, accounts for about 40% of that figure.
Yeah,
But imagine if we could average age of a lymphoma patient, DLBCL, follicular lymphoma, kind of like in the 60s, around 60s to late 70s. If we can extend those patients don't have disease conversion 10, 20 years, they're going to outlive the disease or outlive
that's right.
the possibility of converting.
Yeah,
Yep.
yeah, yeah. Terrific, terrific.
I
Go
will add
ahead, please. I have to tell you, it's so exciting to see you this excited. So go ahead.
one caveat.
Yes.
Long term disease control with these new agents, who's going to pay the medical bill? It's going to be really large. Uh,
Yeah.
um,
Yeah.
you know, CLL, and we'll get to that in a moment. CLL patients are going to be on BTK inhibitors from one to the other to second to third, fourth generation. These are going to be really expensive drugs. They're hundreds of thousands of dollars a year
Yes.
that
Yes.
Medicare is going to have to pay out. Who pays that bill?
Yeah. So a sobering commentary. Yeah, but real. But real. Let's switch to CLL because, the outlook for patients with CLL, the approach is different than it was, 5, 10, 15 years ago. So what was exciting at this meeting?
You know, Chemoimmunotherapy, so-called FCR, BR, bendamustine rituxan, standard of care for CLL patients that goes way back, or chlorambucil way back, right? And when I mean way back, we're probably talking about 10 years. It's still, actually, studies have been done and asking, are you using FCR-BR to treat CLL patients? Just ask clinicians out there. It's still being widely used. Nevertheless, it's very clear now that these so-called BTK inhibitors alone or in combination with venetoclax can control the disease better than chemoimmunotherapy, FCR-BR, and do it for a longer duration. And so those chemoimmunotherapies are outmoded, but getting the word out into the community so people can use these BTK inhibitors in combination is the message that needs to get out there.
Yes.
I want to describe one large study, the so-called Amplify study that was reported by Jennifer Brown. That's abstract 009 for your audience who want to look it up. It's a study of acalabrutinib, so the second generation BTK inhibitor combined with venetoclax versus acalabrutinib, venetoclax plus obinutuzumab, so the second generation rituximab, versus FCR-BR, right? 1 to 1 to 1, all split evenly, and ask, okay, who does best? And the answer is AVO, acalabrutinib, venetoclax, obinutuzumab, or acalabrutinib, venetoclax beats FCR-BR. The 36-month PFS is not reached for AV or AVO, and AV beats out FCR-BR. So the message is acalabrutinib, venetoclax is probably the therapy of choice, at least based on this study. I wanted to add another study looking at which is the best BTK inhibitor, covalent inhibitor to use. You should use ibrutinib, acalabrutinib, zanubrutinib. These things have, for the most part, not been compared head to head. So it's a little bit hard to know. You can basically go by real world data. There was one study by Catal where they actually did that real world comparison. And they ask, okay, of the three BTK covalent inhibitors, who does the best? And the answer is going to be acalabrutinib. Why? So looks like it has better efficacy. Looks like it has less toxicity. The toxicities I'm talking about with ibrutinib are principally grade 3 hypertension, atrial fibrillation, well known with that particular drug, probably due to an off target of ibrutinib, but kind of grade 3 or higher in sort of the 10% range. If you look at it with calibrutinib or zanubrutinib, it's much lower. In fact, in the acalabrutinib venetoclax study that I just talked about, it was considerably low, like around 1%, but it can
Hmm.
be maybe 3%, 5%, statistically different than ibrutinib. That's one factor. The other factor is cost.
Yeah.
That's what this study looked at, going like, well, which is the most economical, efficacious, least toxic? And the answer is acalabrutinib comes out the best. Zanubrutinib's not too far behind. I will say that zanubrutinib and acalabrutinib, in terms of toxicity, are fairly close, and they're distinct from ibrutinib. Acalabrutinib is a little cheaper, and so basically, when you look at it from a cost perspective, efficacy, safety perspective, acalabrutinib comes out the winner. That would be the cheapest alternative for Medicare. But don't forget, from a patient perspective, $2,000 cap, right? Beginning in
Yes. Yeah.
2005. Okay. So who cares? Choose any one of those three. But from the cost of who's paying the rest of the bill, acalabrutinib is the more economical choice for Medicare because you won't have the toxicities that are going to have to be dealt with ibrutinib, for example.
Yeah. Yeah. I want to echo because it's very important that the AV or the AVO, which is more of a biologic approach, targeted approach, is better than chemotherapy.
Yeah, that's the take home message. I mean, like FCR-BR has been outmoded.
Yes.
Um,
Yeah.
but that message has to get, from what I see, has to get out to community physicians.
Yes.
CLL basically can be treated in the community with these agents.
Yeah.
I mean, you have to follow the rules. You have to do venetoclax, has to be step dosed up.
Yeah. Yeah.
So you can't just go in with full dose of venetoclax because you could cause tumor lysis
Right.
syndrome.
Right.
So there's an education about how to use these things. One other comment, just want to talk about, there's the covalent inhibitors, there's the non-covalent inhibitors, pirtobrutinib approved. Now there's going to be degraders, where we simply just we destroy the protein, destroy BTK. There are two companies that have degraders that are reported on at ASH. Those degraders have utility in terms of controlling disease, even in patients that have failed covalent inhibitors for BTK. So that's coming as well.
Let's flip back to talk about more aggressive diseases, mantle cell lymphoma, which I still find to be fascinating. I don't see it much, but in a sense, most patients more aggressive, some patients more indolent. But yeah, what's new in mantle cell?
So, still treated, it's either R-CHOP or some variation of it, is still typically the treatment for a first line mantle cell patient. The question with mantle cell, aggressive lymphoma, right? Going to require therapy, no question about it. The question is, do you need the full 6 cycles of R-CHOP or can you reduce it? And how do you know when to reduce it? This has really been addressed in 2 big studies. One by Martin Dreiling, which was presented last year at ASH, and now another one presented by Fensky with a late breaking abstract. Same question, different endpoints. So the Dreiling work simply said, I'm going to compare what happens if I stop at 4 cycles versus 6 cycles, or something. I can't remember the cycle number exactly. And do I need to go to transplant? Is transplant required to control the disease? What happens if I don't use it in certain patients? And the answer was you don't need transplant for certain patients that have achieved a good clinical, a complete response rate. You just don't need it. So don't use it. The other study by Fensky, the late breaking abstract, asked the same question but used MRD as an endpoint and say, so if your patients are MRD negative, do they still need to go on to transplant? And the answer was you don't need it. And so the result is certain patients will not need transplant for mantle cell lymphoma. And they're both large phase 3 studies done by Dreiling and now by Fensky. I do want to mention one other thing for mantle cell that sort of in the background, but one that LLS has funded and we've been following. So there's another target on the surface of mantle cell patients, to tumor cells. It's called BAFF-R, BAFF-R. Larry Kwok and his colleagues at City of Hope have made a CAR T to BAFF-R. And it turns out there's only been 6 patients treated. 4 of them are mantle cell. 3 out of 4 had a complete response rate in patients that have even failed conventional CAR T therapy. So what I'm saying is there's another CAR T that's coming in the wings for mantle cell patients because CAR T therapy for mantle cell can control the disease, but CAR T therapy, typically we have failures in those patients. We're going to need additional second generation, better CAR Ts, new targets, and here's one of them.
So, and I have to say, from our discussion today, you've really shared that concept that sometimes going against 2 targets may be
Yeah,
better, or adding these
yeah,
therapies to other therapies may be better. So, and the idea of sequencing. So, thank you for adding all that. Let's move on to another very difficult disease, T cell lymphoma, which gets talked about less but is important. So, tell us.
right. So T cell lymphomas, 10% of all lymphomas, rare. And there's multiple types of T cell lymphomas, many different, like about, let's say 10, 15 major types, and they're molecularly distinct. That's a problem because you may have to develop therapies for certain T cell lymphomas, not compared to others. And it's a rare tumor type. So running a clinical trial and trying to find patients that would qualify to show that you really have benefit in certain types of T cell lymphomas is going to be difficult from the patient accrual point of view. Nevertheless, there are targets on the surface that's common to many T cell lymphomas. One is called CD5, another one's called CD7, that is present on multiple T cell lymphomas, including TALL. I'll tell you why that's important in a moment, but also many other T cell lymphomas, leukemias. The TALL patients are important because when you look at ALL and you say patient has leukemia, particularly an adolescent or a pediatric patient, they're usually BALL. Well, BALL can respond to CAR T, blinatumomab, great. For TALL, we really don't have good therapies except for cytotoxics. And by the way, for TALL, 90% of those patients are going to get a good response. However, TALL patients typically have relapse disease, relapse TALL in a pediatric patient, bad outcome. Well, CD5 and CD7 is expressed on the surface of those cells. There's CAR T therapy now to CD7 or CD5 CAR T. The CD7 CAR T, I'm talking about the Gabaldi report. This is out of Washington University, WashU. Um, High complete response rate, 73% complete response rate, promising. The CD5 CAR T is actually being done so far in lymphoma patients, just getting underway. I should add the CAR T therapy, which has really been pioneered in the US, has been explored explosively in China. And there is evidence that CD7 CAR T therapy is going to be useful for patients who have T cell lymphoma. The Chinese have done that actually in large studies. And so it's very likely that CD7 CAR T will play a role in the future in the United States with CAR T therapy. So the CD5, CD7, there's a whole bunch of other targets on T cell lymphomas that we know about that's coming along. I did want to mention there is another antibody that's being developed against certain T cell lymphomas. This was abstract 980. 21 patients, overall response rate 33% as a monotherapy. So that's also coming along. That's Zane et al. It happens to be one of the companies that LLS has been supporting through our venture philanthropy.
Terrific. I'd love to hear some reflections on a career in heme malignancy.
As far as my career goes, I've worked for LLS for the last 11 years. I've been in the cancer drug discovery business for the last 39 years. I started my exploration in medical research back in my undergraduate days doing electrical recording from rabbits responding to different tastes back in the 1972. So I've been doing this business now for about 50 years. 39 years in cancer drug discovery and development, 11 years at LS. I must say, the last 11 years at LS, in looking at all the approvals, there's been about 150 approvals in the last 20 years. But even from 2017 on, 20 FDA approvals just about every year. It has been such a privilege to be here at the right time in the right place, to work at LS to help usher these therapies along. And I will add, having the ability and the access to key leaders in the field have been such a privilege. And to work wide scope at LS to use biomedical research, patient access, and advocacy are three arms to basically attack all fronts to get better therapies and access to patients has been a true privilege. Yes, I am retiring from this job. I call it stepping down.
Okay.
My colleague Laura Grunbaum is taking over, and you'll probably talk to her next year. I am still going to work with LS. There's never enough money to get new therapies. All this work takes millions, billions of dollars to make it happen. And my goal, as it has been really for the last couple of years at LS, to make sure that we have the proper amount of funding to get all this work done, number one, and make sure that the next generation, the next Stephen Grubbs, who we funded back in 93, who treated Emily Whitehead with tozolizumab, first CAR T pediatric patient, is there 20, 30 years later to make sure that those young people who are in the labs in the clinic now are there 10, 20, 30 years from now when we have the further advances. That's the goal. My goal is to provide the fuel for those people to make sure that we're going to get the advances and remain on the same trajectory in terms of improving therapy, or even better than that, which is what LLS's goal is, even improve and accelerate to get increase of life years saved better than what we're even getting now and what we predict is going to happen in the future.
So I have to say that, firstly, so well said and so exciting to hear. I think LLS and patients and families have all been blessed to have your hard work and this career of dedication. And I also want to say, too, as part of and as a leader of this army, is trying to find cures. So, on behalf of myself and our podcast, and I think LLS and a lot of others, thank you.
Been a complete privilege. My pleasure.
Thank you, Lee. I also want to particularly thank Dr. Lee Greenberger, who is the Chief Scientific Officer and Senior Vice President at LLS. Lee, thank you so much for joining us.
My pleasure. A complete privilege to review ASH over the many years.
Again, this is Dr. Ken Miller, and I want to thank all of you for listening to this really terrific, informative, and inspiring episode. And for a list of all of our healthcare professional continuing education activities, podcasts, and healthcare professional resources, please visit lls.org/CE. For any questions or to refer a patient to LLS, please contact our Information Resource Center by calling 800-955-4572. Information specialists provide personalized one-on-one support to help patients learn about their disease, treatment, financial, and support resources. LLS also provides other resources for patients, survivors, and their families that can be found at lls.org/support. I also want to encourage you to sign up to receive notification of future podcast episodes by subscribing at treatingbloodcancers.org. If you have suggestions for future episodes, please click the feedback button on this episode page, and we look forward to you joining us on future episodes.
Thanks for listening to Treating Blood Cancers, the LLS podcast series for professionals. We can be found on iTunes and other podcatchers. You can subscribe at www.treatingbloodcancers.org and provide your suggestions for future topics. Visit our archives section on our website for other great podcasts. Be sure to rate and review us on iTunes. Keep up with LLS by following us on Twitter at LLSUSA and on Facebook at the Leukemia and Lymphoma Society, and access our professional continuing education activities by visiting lls.org/CE. Let's keep the conversation going. Until next time.